Hello everyone. I welcome you all once again to my series of lecture that is Understanding Pharmaceutical Science with Dr. A.J. Hariharan. So today we are going to discuss about one of the important aspects in bacteriology, that is the growth curve of the bacteria. So this lecture I have divided into two parts. First we will understand about the growth curve of the bacteria. Second, we understand about the mathematics, how we can calculate the generation time, and mean growth rate. So first let us understand simply about the bacteria. So bacteria is nothing but it is an unicellular microorganism which is having a size of around 0.5 to 2 micrometer in size, and it has a distinctive prokaryotic cell structure. So it doesn't have an internal organelles which has a membrane limited structure. So the generally if you see the nucleus, that it does not have a nucleus in its structure because the DNA is concentrated at a particular location in the cytoplasm itself, because it doesn't carry a nuclear membrane around it. So it is generally regarded as a prokaryotic cell structure. So today we will elaborately discuss about this growth curve of the bacteria. So to study the growth curve of the bacteria, first let us understand the basic, what is meant by a growth. So if you see for humans, the growth means the increase in the size of a particular organism. Whereas if we consider about the growth of the bacteria, we generally considering about the population size, growth in the population size, not on the individual growth of the bacteria. So generally the population size of the bacteria grows by either a binary cell division of the bacteria or other methods of cell division, maybe such as budding or other types like filamentous type. But most cases the cell division of the bacteria occurs by binary fission. So our study regarding the growth curve of the bacteria is limited only to the increase in the number of cell population of the bacteria, not its individual growth of the bacteria, right? So to study this population growth of the bacteria, we construct a growth curve of growth of bacteria, that is the number of viable cell present after a particular time interval. So for studying this growth curve, we have to perform a simple experiment. So to study this one, what we will generally do is we will take a nutrient broth media, a closed system in a test tube. So to this we will add a fresh culture of microorganism. So this is a batch culture because we are not going to add any fresh media after that addition of the inoculation of the microorganism. So it is a batch or closed system culture. So only thing is we will calculate the viable cells after every particular time interval. So the cell which is present inside the nutrient media, broth media, will divide by a binary fission process. So after each time interval, we will calculate the number of viable cells. Then we will plot a graph. So the graph is plotted with using x-axis the incubation time period, and in the y-axis the log of the viable cells. So this results in a growth curve. So if you see the growth curve of any bacteria, it generally carries the four distinct phases. So these four distinct phases are lag phase, then next it comes a exponential phase, a growth phase, that is a log phase, then a stationary phase, then finally a death or decline phase. So let us understand each phase individually. So first we will understand about the lag phase. So the lag phase generally occurs once a microorganism, in this case a bacteria, has been transferred from the old culture to a fresh cultural medium. So once we transfer it, immediately the lag phase bacterial, immediately it won't start doubling. So once we transfer from a old culture to a fresh medium, it needs some time to adapt to this new condition. So there will be no immediate growth of the bacteria. That means no immediate cell division of the bacteria will occur. So this time period we call as a lag phase. So why the bacteria needs a lag phase? There are many reasons for the presence of this lag phase in most of the bacterial growth curve. The reason one is that So since the bacteria is present in the old nutrient media, so it started losing its ATP energy and enzymes and other cofactors and the protein synthesizing machinery such as ribosomes. So once it has been transferred to this fresh media, so initially it synthesizes all its molecular components and ready for its growth and further cell division. So this time period generally produces this lag phase. And the second major one is that, so initially the bacteria is present in a media which is having a different composition. Then we transfer this bacteria into another media which is having a different composition. So since the change in the composition of the media, the microorganism cannot able to divide immediately. First it has to synthesize new enzymes for to take up these new different nutrients. For example, let's consider a bacteria which is having a ability to synthesize amylase. So once we are growing in a media in which the glucose is present, so since the bacteria the glucose is available, it doesn't produces the amylase, it takes the glucose continuously. So once we transfer to another media in which the carbon source is the starch, so what happens is this bacteria first synthesize the new enzyme amylase, it breaks the starch, then it takes the glucose. So it needs some time period for producing this new enzyme and taking up the nutrients. So this time period generally produces the lag phase. So whatever may be the reasons, generally the presence of the lag phase is simply the time taken for the bacteria to retool itself and makes necessary mechanisms, necessary machineries for the replication of the DNA and to increase its size, mass, and finally it will be in a position to start dividing. So this time period we regard as a lag phase. So what is the general time period of this lag phase? How much time it required for a lag phase? There is no specific time limit for this lag phase. It generally varies considerably from one microorganism, one bacteria to another bacteria. It depends upon the condition of the bacteria as well as the nature of the medium. For example, the bacteria may be present in some very old culture which is deprived of nutrition, or sometimes the bacteria may be in lyophilized form. Once we transfer to the new media, it needs cell retooling. So the time will taken and the length of this lag phase will be generally long. So if, for example, if the culture medium is similar, is chemically different, so still then if you transfer the microorganism, it needs to generate new enzymes to adapt to that new chemical condition. So the lag phase will be long. So if you want a lag phase short, or we doesn't want lag phase, means we need a young, vigorously growing culture, that is the cells in the exponential phase, to be transferred to the fresh medium which is having the same chemical composition. So why this is important? If you think this, the removal of the lag phase is very important for the industrial microbiology experiments. So for example, if you are producing an antibiotics, or if you are producing particular vitamins or an rDNA products in an industry, means we use bacteria for the production process. So once we transfer the inoculum into the production area, if the media composition is different and the bacteria is old culture, then there will be a lag phase. It reduces the productivity time limit of the particular product. So we doesn't need this lag phase. So generally we keep a media chemical composition similar to the production media and helps in this transfer process, right? So the second phase is the exponential phase, otherwise we can call it as a log phase because it produces a logarithmic growth of the bacteria, otherwise we can call it as a growth phase. So you can see a single linear line which shows a exponential phase in this diagram. So the exponential phase or the log phase is generally where the growth will be present and the cell will divide at its maximum full potential. So its growth is basically of these three reasons. One, the bacteria will have its full genetic potential for cell division. Second, the nature of the medium will be comfortably, it has a nutrient availability will be enough for the growth of the bacteria. And third, the condition is also favorable for the cell division, like the temperature, pH, and other condition. Oxygen requirement or vital nutrients requirement will be very high. So the exponential phase will happen. So the cell will start dividing exactly at a particular rate after a particular time interval. So if you see the nature of this log phase, so the rate is constant, rate of growth is constant. So that we call this as a generation time or doubling time. We can see this in the exponential phase. For example, if a bacteria is dividing at a particular time interval, that means it doubles in number in a particular time interval, we generally call as a doubling time. For example, the E. coli will have a doubling time of 15 minutes. That means every 15 minutes a single E. coli bacterium will be divided into two bacteria. So this will, we can see in the exponential phase, and the division will be slightly different, momentum will be there. Thereby we can divide into two types of log phase. One we call as a synchronous growth, and second is the simple balanced growth. So synchronous growth means at 0 minute one cell will be there at exact doubling time. In the case of E. coli, at the 15th minute the one cell will exactly divide into two cell in the 15th minute. Then in the 30th minute, exactly at the 30th minute, this two cell will be divided into four cell. Then after another 15 minutes, that is the 45th minute, this fourth cell exactly at the 45th minute will divide into 8 cell. This type of jumps of growth we call as a synchronous growth, but generally this we can say up to 1 or 2 generations. Apart from that, we never see a synchronous growth. We generally get a rise smooth curve, we call it a balanced growth. So we never find a discrete jumps. So to know the balanced growth, we need some understanding. So in exponential phase, generally the bacteria grow at a balanced rate. It is called as a balanced growth. That means all the bacterial cell constituents is manufactured at the same rate relative to each other. That means all cell will get the nutrients at the same level. It will divide at the same rate. That is called as a balanced growth. That's why the doubling time is exact. Sometimes, generally an unbalanced growth can be seen if a particular essential nutrition is not available to some cells. It will be available to other cells, lead to an unbalanced growth. That means the nutrient levels or environmental conditions changes can lead to an unbalanced growth. For example, let us take the oxygen. Some aerobic bacteria needs oxygen for its growth. So if we supply the oxygen on the surface bacteria will get the oxygen, which is an essential nutrition, it will divide. Whereas the oxygen has a poor ability get dissolved in through the broth media. So the submerged bacteria never gets oxygen. So it divides at a slow rate. So it gives an unbalanced growth. So this unbalanced growth we cannot see in a exponential phase. So generally the unbalanced growth can lead to a stationary phase growth. That means the growth, that is the cell components, doesn't get the nutrients relative to each other. So it leads to an unbalanced growth. So why we have to understand this log phase is that, so since I told in the industrial microbiology, to produce the maximum amount of the products are generally formed in the log phase or the earlier stationary phase. So to understand the log phase is very crucial for the product formation. Also, it is useful for the biochemical and physiological studies also. Then the third phase we call as a stationary phase. So it is becomes a stagnant line. As you can see from the picture, the third one is the stationary phase. So the stationary phase is generally arises because of the nutrition started depleting. So generally, since it is a closed system, it's a batch culture, the population growth will start ceases because of the nutrient unavailability or the toxic formation. The growth becomes an horizontal in nature. And some cases, generally most bacterial stationary phase is reached once the population is 10 to the power 9 cells per ml is reached. This is in most cases. Otherwise, if you add fresh media, then the log phase will start continuing, right? So the reason, if you see the stationary phase, is that the number of viable microorganism will be constant. The living microorganism will be constant. It is because of two reasons. One, the number of cell division will be equal to the number of cell death. So it will lead to a stagnant. Otherwise, the cell stopped dividing, but it is surviving. That is, it is metabolically active, but it doesn't go for cell division. It's only surviving. There also we can see the total number of viable cells will be stagnant. So this leads to a stationary phase. So the reason I have already told you, if you see, the major reason is the nutrition limitation. If the nutrients is not available, the growth will lead to the stationary phase. But in continuous culture, the fresh nutrients will be supplied, the log phase can be maintained. And some cases, certain essential nutrients will be depleted, thereby the growth of the rate can be drastically decreased. So one example I have already told is the oxygen. So the aerobic organism needs oxygen. So the oxygen availability is very important for its growth. So the oxygen has less solubility into the media. So what happens is the surface bacteria will grow at a faster rate, whereas the submerged bacteria doesn't get adequate oxygen concentration. So to maintain the log phase, what we generally do is that we shake or aerate this bacteria to keep in the log phase. So if the oxygen is completely deprived, means it leads to the stationary phase. The second major one is the toxic waste, which is also leads to the stationary phase, because the toxic products will started accumulating. This will causes death to some bacteria. So the number of cell division will equal to the number of cell deaths. For example, streptococci will produce certain acidic products like lactic acid or certain organic acid once it comes into the food, such as sucrose or glucose. And as through glycolytic pathway, it forms this lactic acid as an end product. So this reduces the pH of the media and produces an unfavorable growth condition to the bacteria. Thereby the stationary phase can be reached. And the final one is the nutrient supply, which is the very important one. Once the major nutrients like the carbon source, the sugar supply is limited, the growth will stop. So apart from this, the last one is, even though the nutrients, the essential nutrients, favorable environmental, everything are there, some bacteria goes to the stationary phase once it reaches a critical population level. So it's a rare condition. So these are the major ones which leads to the stationary phase. And the final one is the death phase or decline phase, or we also call as senescence. So you can see that the graph started falling down. This is called as a death phase. So it is also called as senescence. So the number of living cells will start a decline in a particular phase. We generally record as a death phase. So in this, we call the death of the cell is that it is metabolically inactive, and it is inactive of cell division, only we call as death. Generally, if we consider what it means by a bacterial death, is the lysis of the cell. The bacteria, we call it is died once the bacterial cell got lysed. So in this case, we not considering the lysed cell as a death, we only considering the metabolically inactive, and it is non-viable in, not ability to self-divide or non-viable in nature, we call as a death of the microorganism. The major reason for this death is the death phase is the detrimental effects, environmental changes that is happening. So one, the nutrition is depleted. So no essential nutrients or other nutrients or nothing is available to the bacteria for its growth. So eventually it cannot survive even in the stationary phase. It started dies. And second, the buildup of the toxic waste will cause us the death of the bacteria. So it finally leads to loss in its viability, and finally death occurs. So these four are the major phases of the bacteria: lag phase, log phase, stationary phase, and death phase. So by studying this various growth phases of the bacteria, we can come with certain calculation how the growth of the bacteria is happening, which is very important for understanding the growth characteristics of the bacteria as well as for the manufacturing of certain pharmaceutical or biotechnological products. So for understanding this one, let us give a brief understanding about the mathematical concept based on the growth of the bacteria, how we can mathematically calculate the growth of the bacteria. So one important thing is the generation time or doubling time. That means every particular time interval, the cell can double in its size. So this we can see in the exponential phase. In the log phase, what happens is every particular interval of time, the cell will double in its size because of the continuous cell division which is happening in the exponential phase. So the population will double exactly at the specific length of time we generally call as doubling time. So it is regarded as a generation time. So one generation happened. After that, the same time interval, the second generation will happen, and similar interval, the third generation will happen. So this I have explained with a simple illustration. So let's consider with a simple example. It is a hypothetical concept. Let's consider we have taken a broth media in a test tube. Inside it, I have inoculated a single cell. So this single cell is going to divide exactly at 20 minutes. So if you put this into a table, so I have put a table. So in this table, the time, so in time 0 minutes, I have told every 20 minutes the cell will divide. So we have kept 0, 20, 40, 60 like that. So number of division, so at 0 minute there is no cell division will be happen. So we have a formula called 2 to the power n. So 2 regards the doubling time, n is the number of cells. So in this, at the initially, if you see, there is n is the number of generation. So at 0 there is no generation. 2 power 0 is equal to 1. That means I have inoculated a single cell. There is no generation happen. So the n0 is the initial population size. At 0 minute I have only one cell. So 1 into 1 is the 1. So log to the power nt, that means at 0 minute is nt is becomes n0, so 0 becomes 0, right? So after 20 minutes, what happened? The first cell division occur. So 2 to the power 1, one generation, that is 2 cell, that is one cell has become 2 cell. So the initial population n0 is 1. So since I have inoculated only one cell, 2 power 1, that is 2, 1 into 2, so we have 2 cells. So log 2 to the base 10 is 0.301. So after 20 minutes, that is the 40th minute, the second generation happened. So 2 into power 2, that is 4 cells. So 2 divided into another 2 cells, we got 4 cells. So initial population is 1 into 4, we have 4. So this is the n to the power 0. So similarly, we can get many things. So up to various time intervals, we will get this one, right? So out of this, if we do practically, I can get only two values. What I can get is nt. After 8 hours, what is the total population? So I also know at 0 minute what is the initial population. This generation time, mean generation time, mean growth rate, everything we can calculate later on through graphically or mathematically, right? So this I will tell you, right? So in this experiment, what we have seen is at 0 minute there is single cell. The doubling time, generation time is 20 minutes. So after 20 minutes, we got one cell divided into 2 cell. After 40 minutes, this 2 cell divided into 4 cell. And after 60 minutes, this 4 cell divided into 8 cell. Like that it started going on. So what happened is, every particular time interval, this single cell becoming a 2 times of the cell. So the population at the doubling of every generation increases by 2 to the 2n. So this we can show with a simple formulation. N is the number of generation. So 2 to the power n, that I have shown in the picture. So if you see this exponential microbial growth, so for 6th generation, we have drawn a graph. So in this graph, you can see that. So if you plot a graph between the time against the number of viable cells, we got a parabolic curve which is not, we cannot used for mathematical calculation. So if I apply a logarithmic characteristics, I will get a linear curve. So out of this, I can easily calculate the generation time. So generally the logarithmic form is applied for the mathematical calculations. For 6th generation, both normal graph and the logarithmic graph you can see from this picture. From this directly we can calculate the generation time also. Let us start some calculation part, how we can calculate this generation time, mean generation time and mean growth rate. So let us calculate NT, that is the number of cells at time t is equal to n0 into 2 to the power small n. So n0 is the initial population, 2 is the doubling time, n is the number of generations. So this I have told, n0 is the initial population number. If the initial population is 1, after 1 generation, 2 to the power 1, 1 into 2, we have 2 cells. That is the NT, right? So NT is the population at time t, n is the number of generation. So this is the basic formula we are starting up. That is the NT is equal to n0 into 2 to the power n, right? So let's take the logarithmic form. Since the normal form will have a parabolic curve, for mathematical calculation we need to a logarithmic form. So if I put the logarithmic form, so I have put log into both the things. So log nt is equal to log n0, so plus we have 2 to the power n, so log 2 to the power n, so the n can be bring it down, n dot log 2. So what I am doing is I need to calculate the generation time. So we need the generation time. So since we need nt and n0, so we have to calculate the generation. So I keep the n this side. So this n0 will go that side. So its plus symbol becomes a minus, log nt minus log n0. So n dot log 2. So log 2 I bring it to the down. So n generation time, n is equal to log nt minus log n0 by log 2. So the value of log 2 is 0.301. So n is equal to log nt minus log n0 by 0.301. So next, this is one part, calculating the generation time. Next is the mean growth rate. So rate of growth, what is the rate of growth at every particular unit of time? So this we can calculate by a simple formula, k is equal to n by t, right? So k is the rate of growth is equal to n per unit time t, right? So k, instead of n, we can put the formula which I have derived it in the previous equation. So n is equal to log nt minus log n0 by 0.301. So we can substitute in this mean growth rate. So if I put in the mean growth rate, so log nt minus log n0 by 0.301, the t will be present like that. So the generation time formula is log nt minus log n0 by 0.301. The mean growth rate k formula is log nt minus log n0 by 0.301 t, right? So let us consider that the mean growth rate, that is the mean generation time for every hour. What is that? We can calculate it. So the mean generation time, that is the time taken for the population to double in its size exactly. That is the mean generation time or mean doubling time. So instead of n, which is generation time, for mean we can take as a g. So when t is equal to g, then it causes a mean generation time. So let us consider nt is the time that double its population. Nt is equal to 2 times of the initial population. That is becomes the mean generation time, right? So we can substitute this into the equation, that is the generation time equation. So we have the mean growth rate k is equal to n by t, that is log nt minus log n0 by 0.301 t. So instead of this nt, we can put 2n0, that is the mean growth rate. Okay, so 2n0, if I put log 2n0 minus log n0 by 0.301 g, so log 2 plus log n0 minus log n0. So this plus log n0 and minus log n0 will cancel. Similarly, log t and 0.301 will also be cancelled. So the mean growth rate is the inversely proportional to mean generation time, that is g, k by 1 g, right? So the mean generation time is inversely proportional to the mean growth rate that we have got from the equation. So we can calculate this mean generation time directly from the graph. Also we can calculate from the equation. So first I will tell you how we can calculate from the graph. So let's consider we have inoculated a bacteria, then it shows a particular growth, then we will calculate a mean generation time. How we can do it from a graph, right? So in this graph you can see that after the log phase we have to calculate the mean generation time, that is G. So the initial population, if you see, it is 0.050, right, at a particular time I have calculated. Then 0.5 cells gets doubled, that is 1.0, right? So these 2 points I have marked. This is the initial point 0.50 and this is the second point, that is 1.0. So the time between these two things is the G. So that I have calculated directly. So in this we can see that how we can calculate the mean generation time with this graph. So the 0.05 is the value. So we have put it at the time we have calculated it. Then its double number of cell is 1.00. 0.5 into 2 is 1.00. So we have marked in the exponential phase and we can directly get from the graph the mean generation time. So the graph is plotted between the population data in the y-axis, logarithmic population data in the y-axis, and the time in hours in the x-axis. So we can directly get the mean generation time. So the time taken to double of its population. So the population has been doubled from 0.5 to 1. That is how much cells, how much time it has taken. It's directly read from the graph itself. So whereas this can be calculated through equation also. So suppose the initial population is 10 to the power 3 cells, that is 1000 bacterial cells per ml. Then after 10 hours the population is 10 to the power 9. Of this the N0 is 10 to the power 3. The NT, that is at the time T, that is 10 hours, the population is 10 to the power 9. If I put in the formula, that is the mean growth rate K, the formula is log 10 to the NT minus log N0 by 0.301 into G or T, right? So the K log 10 to the power 9, which is the NT, and initial population N0 is 10 to the power 3 and the T is 10 hours. So 9 minus 3 by 3.01 hours. So we got the mean growth rate is the 2 generation per hour. So in 1 hour there is 2 generation, whereas the generation time G is K is equal to 1 by G. So G is 1 by K. So 1 by 2.0 is equal to 0.5 hours per generation. That is 30 minutes per generation we can get calculated it. I think this lecture will be very helpful for you understanding the bacterial growth and the method of calculation of mean generation time and mean growth rate. So thank you very much. So continuously view my various lectures on this channel. So thank you very much.